Students, welcome you all on the behalf of Rahul Anand Tutorials. So as you know that I am Rahul Anand and this is somewhat on my channel whose name is actually the Rahul Anand Tutorials. So now let's begin with the very interesting topics which are often being asked in terms of your need, often the Aiims, often the All India Pre-Medical Examination. So these are literally belonging from which of the chapters of your book? So these are belonging from the cell, the unit of life. Now, so when we are talking about these cases in terms, so today we are going to study about what? So today we are going to study about the lysosomes. So when we are talking about the lysosomes, so these are what? So lysosomes are generally being found in terms of what? In terms of the eukaryotic cell. As well as when we are talking about these cases, so these are often being abounded with reference to what? With reference to the red blood cell. Fine, in terms of the animals, often. So when we are talking about this case in terms of the red blood cells, so it will be generally being found with reference to what? With reference to the animal body. Remember? So particularly having the three broad types of the quantity as well as the resemblance in terms of its name only, such as what? Such as the red as well as the blood. Fine. So blood components will be generally being present in terms of the animal's body. Plant is having the another type of the components, but not this red blood cell. Often you are very clear about that, means what are actually the eukaryotic cell. What are that? So the cell which is having the defined type of the nucleus as well as the other cell organelles as well as what other or ingredients with reference to the cell. So this will be coming under what case? So this will be coming under the eukaryotic cell. Often when we are talking about these cases in terms of the lysosomes, so these are particularly often having what? So these are often having the certain types of the exception case, such as what? So this will be not present with reference to what? With reference to the prokaryotic cell. So you are very clear about that. Means what are actually the prokaryotic? So when we are talking about the prokaryotic cell, so you are very clear about that. Means what are actually the prokaryotic cell? So the cell which will be having the resemblance as well as what? In which there will be the absence of what? Of the nucleus as well as the other cell ingredients or other the organelles. These will be coming under what case? So this will be coming under your prokaryotic cell. So now let's proceed with reference to the another facting. So when we are talking about the lysosomes, so these are what? Now let's understand about these, means what are actually the lysosomes. So lysosomes are what? So lysosomes are the thin membrane vesicles which will be having what? Which will be having the different types of the digestive enzymes. So you are very clear about that. Means what are actually the digestive enzymes, such as what? So we will be having the protease, then what? Then the carbohydrate, often the lipase, as well as what? As well as the phosphatase. So these are actually what? So these are the different kinds of the lipase with the reference. Often these are what? So these are actually the enzymes with reference to our digestive enzymes. Fine. So when we are talking about the case of the protease, so it will be directly proportional for what case? So your protease will be directly proportional for the protein case. And you are very clear about that, means what are actually the protein. Tell me. So protein are the macromolecules of the amino acid, often being further divided in terms of the four broad categories, such as what? Such as the primary structure, the secondary structure, the tertiary structure, as well as the quaternary structure. After that you are having the carbohydrate. So carbohydrate are what? Carbohydrate are the optically active polyhydroxyaldehyde as well as the polyhydroxyketone, often being divided in terms of the three broad categories, such as what? Such as the monosaccharide as well as the many types of the disaccharide as well as what? As well as the oligosaccharide. So monosaccharide, then polysaccharide as well as the oligosaccharide. Remember? So the carbohydrate which cannot be hydrolyzed or either broken down in terms of the simpler units, coming under what case? So it will be coming under your monosaccharide case. Often after that you are having the polysaccharide. So the carbohydrate which could be broken down into what? Into the simpler sugar with reference to how much of the ratio? With reference to the 2 to 10 units of the simple sugar. Coming under what case? It will be coming under your many terms of the polysaccharide. After that you are having what? The oligosaccharide. So the carbohydrate which can be hydrolyzed with reference to what? With reference to more than 2 to 10 units of what? The simpler sugar, coming under what case? So it will be coming under the oligosaccharide case. So these are coming under primarily coming under from the carbohydrate case. And after that you are often having the lipase, directly proportional coming from the what case? So these are coming under from the many types of the lipids. Remember about the case of the lipids? So lipids are actually what? So the lipids are the natural occurring what? So these are the natural occurring organic substances which are particularly insoluble with reference to the water molecules, as well as having the influencing role in order to provide what? In order to provide the particular strength with reference to the cell body, often the different types of the cell ingredients. Understood about the role of the lipids? So these are particularly coming under the lipase. Fine. So when I am talking about the lipids, so this will be what? So these are quite a bit the kind of main term of what? The organelles of the cell. Often you are having the nucleolus. After that you will be often having the nuclease. Fine. So you will be after that you will be often having the nucleates. Nucleates are directly proportional for what? So these are directly proportional for the nucleus case, and you are very clear about that, means what are actually the nucleus. So the nucleus will be having what? So the nucleus will be containing the chromosomes, and chromosomes are known as what? So these are known as the vehicle of heredity. Fine. So these nucleus are what? So these are the cell organelles as well as the cell ingredients with reference to what? With reference to our cell. As well as when we are particularly adding the EASE, so this will be coming under, or either the A's. So this will be particularly considered as what? So this will be considered as the enzymes case, such as in terms of the protease, then the lipase, as well as what? Often the case of the carbohydrate. Fine. Often the here, many terms the phospholipids as well as the phospholipids. Now, after that you will be having what? After that we are having the case of the enzymes. So you are very clear about that. Means what actually the enzymes? Tell me. So enzymes are the biocatalysts, playing important role in order for what? Playing important role to increase or decrease the rate of reaction without being altered. Remember? So we'll be often having the different types of the enzymes in terms of the protease. Coming under what case? The protein case. Often your enzymes are further being divided in terms of the two broad categories. So these are what? So these are the apophyters as well as the coenzymes. So coenzymes as well as what? As well as the apophyters. One will be the proteinous enzymes as well as on the another hand. One will be what? So it will be the non-proteinous enzymes. Remember? Now, so when we are talking about this case in terms of the lysosomes, so these are particularly discovered by whom? So that, and after that, when we are talking about the case of the lysosomes, so these were particularly discovered by whom? So it was discovered by the C.D.U. Doof. Particularly in terms of what of the era? In terms of the 1955. So when we are talking about these case, means the C.D.U. Doof was actually the scientist, played an important role to discover, as well as he particularly named the lysosomes with reference to what of the era? With reference to the 1955. Due to the presence as well as what? So due to, because it were actually resembling the structure as like what? As like the hydrolytic enzymes. Thus it was given the name of what? So thus due to these factors in terms of what? In terms of, because it is resembling the hydrolytic nature as well as the hydrolytic enzymes nature, it was given the name of what? The lysosomes. Now, so let's understand about these, means what are actually the structure of the lysosomes. So these will be particularly having the resemblance as well as the structure. Find here. So these are often having the highly viscous fluid structure. So find here the structure of the lysosomes here. So these are known as the unit membrane. After that you are having the highly viscous fluid. Now, so when we are talking about these cases in terms of the lysosomes, so this will be often having the different as well as the vital types of the components as well as the parts in it. So you could often find here on the board here. Thus, when we are talking about the structure of the lysosome, so it will be resembling what kind of the structure? So these are particularly resembling the single unit strain type of the structure. Often, when we are talking about these cases, so this will be particularly having the globular structure with reference to of how much of the diameter. So it is actually around 0.2 to 0.8 with reference to the μm in terms of the diameter. So μm is actually what? So these are the unit with reference to your diameter case. Now, so when we are talking about the shape as well as the size of what? Of the lysosome. So these are quite a bit what? So these are quite a bit the pleuromorphic in nature. Means what? So your shape and size with reference to the lysosome. So these are quite a bit pleuromorphic lysosomes in nature. Thus, these are often known as what? So thus these are often known to have the resemblance with reference to the pleuromorphic nature. Now, so when we are talking about these cases, so you are often having the broad kind of the thing in it, such as what? And when we are talking about the case of the lysosome, so these are often known as what? So in the acidic medium, particularly all the hydrolytic enzymes particularly become active. Thus these are known as what? Thus these are known as the acidic hydrolytes. So you could often find here on the board here. Now, after that, when we are talking about the case of what? Of the lysosomes with reference to what are actually the role of the lysosomes. So these are particularly playing important role in terms for what? So these are playing important role in order for engulfing as well as digesting the waste material, which are quite a bit hazardous as well as what? As well as particularly what? As well as particularly dangerous with reference to the healthy status with reference to our cell. Now, so when we are talking about the case of the lysosomes, so these are often known as what? Tell me. So these are known as the suicidal bag. Why is it so? So primarily when we are talking about the lysosomes, you have to understand the particular resemblance as well as the structure in it, such as it is having the unit membrane structure playing important role, as well as when there will be particularly any kind of the here interaction with reference in terms of the healthy status with reference to your cell. Thus after that what will be happening? Thus after that here minute of the lysosomes wall or other what? The thick membranous structure or other the unit membrane structure particularly break, and after that what will be happening? And after that the hydrolytic enzymes will particularly escape from here minute of the lysosomes in terms of what? In terms of the break. After that what will be happening? So your hydrolytic enzymes will particularly escape in terms of the cytoplasm. Thus because of these are known as what? So your lysosomes are known as the here minute of the suicidal bag. Often when we are talking about the case, means what are actually the here minute of the cytoplasm. Remember? So these are what? So these are the fluid viscous substances with reference found in terms of the what? The cell organelles, as well as what? Around 80% of these fluid, means what? These fluid ingredients with reference to your cytoplasm are generally having what? Are generally having the H2O molecules with reference to the water molecules. Often most of the cell ingredients are generally being present with reference to what? So these are particularly being merged in the other cell organelles with reference to in these only, meaning in terms of the cytoplasm. Often when we are talking about the lysosomes structure, so these are often known as what? So these are not known as the unit membrane structure. Often these are what? So these are particularly very micro when we are particularly comparing with reference to the another component of the cell, such as we are having the nucleus, the endoplasmic reticulum, the Golgi complex, the Golgi apparatus, then the vesicles. Often you will be having the nucleolus, remember, as well as the vesicles, as well as what? The rough endoplasmic reticulum, as well as the smooth endoplasmic reticulum. Now, so we are talking about these cases in terms of what? In terms of the here, in terms of the types of the lysosomes. So these are oftenly being divided in terms of the two broad categories. So these are what? So these are the primary lysosomes as well as the secondary lysosomes. So when we are talking about these cases in terms of the primary lysosomes, so these are what? Here. So in terms from the what? So from the Golgi complex as well as the Golgi apparatus. These are particularly being released in what? In the vesicles. So when we are talking about these cases, means what are actually the Golgi complex. So you are very clear about that. Remember? So Golgi complex are as well as when we are talking about these cases in terms of what? The types of the lysosomes. So you are very clear about that. Means what are actually the types of the lysosomes. So these are what? So these are the here main term of the primary structure or either the primary lysosomes as well as what? As well as the secondary lysosomes. So in this case, what will be happening in the case of the primary lysosome from the Golgi apparatus as well as the Golgi complex? These lysosomes will be particularly released in terms of what of the form? In terms of the form of the vacuoles. Remember about the case of the vacuoles. Now, as well as when we are talking about the secondary structure of the here in terms of the lysosome. So these are what? So when particularly your food ingredient as well as what? As well as the primary lysosomes generally combined, it result in the formation of what? So this result in the formation of what? The secondary lysosomes, or you can say that when the particular food particles or rather the food ingredients generally fuse with reference to the primary lysosomes coming under what case? As well as the result obtained will be what? So this will be coming under the secondary lysosomes. As well as when we are talking about these cases, means what are actually the Golgi apparatus as well as the Golgi complex. So these are actually what? So these are the same name, such as we are often having the fluid moistic model of what? So fluid moistic model of the plasma membrane. So these are often known as what? So these are often known as the proteinous model of the plasma membrane. So we are talking about the case of the Golgi apparatus as well as the Golgi complex. So these are particularly being demonstrated as well as discovered by whom? So it was discovered as well as demonstrated with reference to what? With reference to the Camilo Golgi. Often when we are talking about the case, so these structures as well as meaning of the Golgi complex or rather the Golgi apparatus are generally being found with reference to what? With reference to the eukaryotic cell. Often these are particularly being found as well as present in terms of the animal cell. Often these were particularly discovered as well as found in terms of what of the structure. So these are particularly being viewed with reference to the electron microscope as well as these are often particularly having the different types of the vital types of the resemblance as well as such and when we are talking about the case of the Golgi complex as well as the Golgi apparatus, so you are often very clear about that. So it was particularly being discovered by whom? So it was discovered by the Camilo Golgi. Often when we are talking with reference to what? So he performed the experiment as well as found these minute of the Golgi complex as well as the Golgi apparatus with reference to what? With reference to the nerve cell of owl. Often when we are talking about these cases, minute of what? Minute of the Golgi complex as well as the Golgi apparatus. So these are viewed with reference to what? So these are particularly being viewed with reference to the electron microscope. Often these are actually what? So these are particularly resembling the dense strain reticulum structure, which are particularly being viewed with reference to what? With reference to the electron microscope. As well as when we are talking about these cases, so these are generally particularly present where? So it will be present as well as found in terms of the eukaryotic cell. Often you are having the exception case. So these are what? So these are not being found with reference to the red blood cell in terms of the animal. So primarily when we are talking about the red blood cell, so it will be present where? So it will be only present with reference to the animal cell. Example, as well as understood about the meaning, here meaning of the red, as well as the blood, as well as the cell. So in the plants we are not having these kind of the resemblance meaning in terms of the blood. Often when we are talking about these cases, so what are actually another exception? Tell me. So we are not having this type of the resemblance within term of the sperm cell of the bryophytes as well as what? As well as the here meaning of the pteridophytes. Often when we are talking about these cases, means what are actually the another exception. So this will be not found with reference to what? With reference to the here meaning of the prokaryotic cell. Remember what are prokaryotic cell? So the cell which will be not having the here meaning of the nucleus in terms of the defined ratio or another type of the cell ingredients as well as the organelles coming under what case? So Tell me. So it will be coming into the prokaryotic cell. As well as when we are talking about these cases in terms of the Golgi apparatus. So these are particularly resembling the different types of the vital structure as well as in terms of the plant body. It will be often found in terms of what? In terms of the Golgi complex as well as the Golgi apparatus. So these are being scattered. Thus these are known as what? Thus, due to the resemblance in terms of the scattered, in terms of the plant body, these are known as the diatosomes. Often when we are talking about the structure in terms of the Golgi complex as well as the Golgi apparatus, remember? So it will be having the cysteria, then the tubules as well as the vesicles. So what are cysteria? So cysteria are the flattened membrane, sac-like structure. Often when we are talking about the case of the tubules, so tubules are what? Tell me. So tubules are what? Tell me. So from the peripheral area of the cysteria, the tube-like structure are generally being arising. So these are considered as well as known as what? So these are known as the tubules. Often in the last you are having what? So in the last you will be having the here. So you have read about the cysteria, then the tubules, then the vesicles. So what are vesicles? So vesicles are the round, then the oval type of the structure. Generally being found with reference to what? So these are particularly being bounded with reference to the here, merit of the budding of the hydra. Budding of what? So budding with reference to the vesicles case, it will be result in the formation of what? So it will be result in the formation of the and when we are talking about the case of the vesicles, remember about their structure? So their structure is particularly being somewhat like the round as well as the oval structure. Often, as well as when we are talking about the major components as well as the role of the Golgi complex as well as the Golgi apparatus, playing important role for what? Playing important role to pump the waste material with reference out of the cell to maintain what? To maintain the healthy status with reference to the cell. Remember? So cell are what? So cell are the here main of the structure and functional unit of our life, or either the functional as well as the structural unit of the lifeline. Often when we are talking about these cases, we need of what? We need of the Golgi apparatus. Important role. So these play important role to secrete what? To secrete the hormones as well as the another type of the enzymes. Fine. So when we are talking about the hormones, so these are what? So these are the essential type of the what? The essential type of the biomolecules, literally playing important as well as the influential role to maintain what? To maintain the life status as well as the healthy status with reference to our body. Often in this case, you will be having what? The endocrine glands as well as the exocrine glands. So one is directly proportional for the ductless gland, and another one is what? So another one is directly proportional for the ductful gland. And in last you are having what? The enzymes. So what are enzymes? So enzymes are the biological catalyst playing important role to increase or decrease the rate of reaction without being what? Without being altered. Overall, the course of reaction. Remember, often being divided in terms of the proteinous enzymes, the non-proteinous enzymes. One will be what? One will be the cofactors as well as the one will be non-proteinous enzymes. And you are very clear about that. Means what are actually the proteinous enzymes. Often when we are talking about the another role of what? Of the Golgi apparatus as well as the Golgi complex. Playing important role for what? Tell me. So these are playing important role for here. Minute order for transporting the here. Minute of the another type of the packaging as well as transporting the particular ingredients as well as what? The substances which should be transported with reference to what? Which should be transported with reference to the minute of the intracellular spaces. So you are very clear about that. Means what are actually the intracellular spaces? Remember as well as understand. So these are nothing else but the cytoplasm as well as the another ingredients in which what will be happening, in which our particularly cell ingredients as well as the cell organelles are generally being planted as well as generally being found. Often, in terms when we are talking about these cases in terms of the final as well as the here very important role of the Golgi complex as well as the Golgi apparatus, playing important role for what? Playing important role for the synthesizing of the lysosomes. And here we have just studied about what are actually the lysosomes. Remember, so lysosomes are known as the suicidal bag, having the complex type of the structure with reference to the unit membrane structure. So these are often having the pellucidal structure. Thus, these are known as what? So these are particularly known to have the pelludomorphism in terms of the nature, as well as we have often understood about the different as well as the vital cases in terms of this, in terms of the lysosomes. Thus, this is somewhat the condition when we are talking about the case of the lysosomes. So you could often write this. So if you like the work with reference to the education as well as the lectures as well as the videos, so you could often subscribe, like as well as comment below in the text as well as what? As well as share my lectures with reference to what? With reference to the Rahul Anand tutorial. So I thought you have got the concept. So please like, subscribe as well as share my lectures with reference to what? With reference to the Rahul Anand tutorial. So very thank you.